Hello, and welcome to this webinar titled Host Cell Protein Characterization and Mass Spectrometry Strategies, hosted by Biopharma Asia Magazine and presented by Xuezhi B, Group Leader, Proteomics, and Senior Scientist at Bioprocessing Technology Institute, A*STAR. My name is Stephen Edwards, and I'll be your moderator. Now, please allow me to introduce our presenter. Dr. B has more than 25 years of research experience on proteins and proteomics, working on samples ranging from rice, dust mites, fungus, virus, tears, sweat, cancer cell lines and tissue, embryonic stem cells, platypus venom to CHO cell-generated biotherapeutics. He obtained his PhD in the year 2000 from Wuhan University, followed by postdoctoral training at the International Rice Research Institute and National University of Singapore. He joined A*STAR as a research scientist, where he contributed to the chemical proteins for chemical probe drug target identification, protein PTM, and interacting partners characterization. Currently, he's leading the proteomics group at the Bioprocessing Technology Institute, focusing on mass spectrometry-based therapeutics product CQA's characterization, identification, and quantification of HCPs and aggregate associate proteins, and proteomic profiling of upstream and downstream CHO culture processes by LC-MS. Recently, his group is actively collaborating with multiple industry partners to develop high-performance LC-MS solutions for HCPs analysis and to profile MAB-producing CHO cell culture processes. I will now be handing over to our presenter. Welcome, Dr. B.
Okay, thank you, Stephen. So I'm happy to present today with the whole cell proteins and the characterization and the mass spec strategies. Okay, here is first one I would like to introduce of my institute. Whereas the Bioprocessing Technology Institute is an academic research institute. It's funded by the government agency for science and technology research. BTI has the key capabilities in 6 areas: immunology, antibody, and cell culture technologies, and as well as the downstream processing, glycoanalytics, and omics technologies. With these capabilities supporting 4 of major BTI's core competence in the areas of biologics, we work on mainly on the molecular antibodies, proteins, peptides, as well as the RNAs. As well as in the cell therapy area, we work on the CAR T cells, embryo stem cells, and mesenchymal stem cells, as well as the exosomes. With the microbial biotechnologies, work on highly with enzymes, highly valued molecules. With the medtech instrumentation, we work on most of these microbioreactors. As a monoclonal antibody is currently considered one of the major classes of the biopharmaceuticals in development. The main specifications of these molecules continue to be the majority of the Chinese homocysteine cells. The core competence developed by DTI since its first inception in biological development. Here we have built a strength in almost all aspects of the biopharmaceutical value chains, from discovery to cell line development to upstream, downstream process development to product and process analytical and omics technologies. So with the technology, with the bullet points are our in-house technologies, in such as impact, targeting integration of the cell line development, as well as the chromatin extraction technologies in downstream. With this kind of competency, we aim to develop lawful technologies as well as to first facilitate biologics development and manufacturing in Singapore and beyond. Today's topic is the whole cell proteins. The whole cell protein is required to monitor and report by the regulatory agencies such as the FDA, EMA, and also International Conference Harmony guidelines. Host cell proteins is the process-related impurities. Proteins, this is the source from proteins secreted from host cell along the drug product. During the recombinant protein expressed as a cell stress, the aberrant protein trafficking also circulated to the media, as well as the cell lysis during the cell growth apoptosis. Under the downstream process, product associations, especially during the protein A loading, with strong attractive interactions with monoclonal antibodies of the host cell proteins as well. And also with the monoclonal antibodies during co-elution with the monoclonal antibodies on the polishing chromatogram media. So if we don't remove the host cell proteins from the final product, it impact and or affect the drug's efficacy, such as the binding capacity of the biological activity, and also the stability due to enzymatically activity of the host cell proteins, especially, and the most important is the safety of the patient. If the host cell protein has some immunogenicity, and also it will cause direct in clinical effect of the immediate toxicity, allergic reactions or side effects. Cases of the host cell protein caused clinical hold or delay of drug development and the productions. There's a number of the examples from such as the recombinant factor lying and also the human growth hormones, antibody drugs of anti-IR13 and also fusion proteins CTLA4A4 For the safety of the patient and product efficacy, as well as the manufacturing and process consistency, we need to do real-time or in-process testing for one of the HCP as the critical attributes during the various stage of throughout the manufacturing process and the release testing for the final drugs. To remove host cell proteins, multiple purification steps are involved in the downstream process due to diverse immunological and also physiological properties of HCPs. General downstream process monoclonal antibodies usually carry out by protein A affinity purification followed by several chromatography polishing steps such as the exchange, strong catch exchange and a strong anion exchange to final drugs. However, clearance host cell proteins is a challenge task due to the affinity between host cell proteins and the products, leading to corrosion, co-elution of such kind of the hitchhiker host cell protein HCPs during purification steps. The detection of host cell proteins is also complicated by the heterogeneous composition of host cell proteins, which is varies according to the host cells, cell viability of products, current culture process, as well as downstream purification techniques. Indeed, we are in-house with collaboration with our downstream colleagues. We have identified some of the chromatins as the cause of the molecular antibody aggregates and HCP co-elutions with the host cell protein, molecular antibodies during protein A purifications. Once we apply the chromatin extraction technology with in-house, can significantly clear the host cell protein, thus able to reduce the poly steps from 2 steps to 1. Furthermore, profiled host cell protein change during the manufacturing process from a high level of to the final clean product. There's a really significant dynamic range from the very high concentration of host cell proteins in the original material before clarification, so to the final products, really the single digit ppm. There's a heavily dynamic range difference between the product and host cell protein, more than 5 orders in general. some problem with this. So there's a major challenge of the protein, the characterization host protein in the lower band of HCPs in the final products after downstream purification process. For the strategy of host cell protein analysis, there are two major orthogonal methods. First one is the immuno-based assay, and another one is the mass spec-based method. However, the immunoassays is really well established, the gold standard to measure HCP proteins sensitive up to 1 to 100 ppm is the general guidelines of the acceptable range during the ICP Q6B. The one is more robust, well established, and also high throughput can be automated, and also it is. But the drawback is the highly dependent on the quality of the antibodies. And also unable to identify and quantify an individual host cell proteins. The major concern of the immunoassay is the coverage of the antibodies. Normally, they difficult to really detect weak and non-immunogenic HCPs, as well as the jackpot effect. A few HCPs with high affinity antibodies dominate the signals, while low affinity antibodies probably have the really minimal impact of the total readings. Antibody-bound protein and co-purification HCPs are difficult to detect by antibodies. And also the process-specific support process development, the subset of HCPs may change during process development. So with the development of different assays to be slightly different, keep changing, the time consuming, and using the platform assays versus process assays to be applied. To deal with some of the demonstration of the coverage, and then currently using the 2D Western blot, using 2D, but this still detection limit depends on the quality of the antibodies. Coverage also limited by the separation of the hydrophobicity and the low abundance of proteins, and compared to the high epsilon range. And also especially the high PI range and the low molecular status coverage of the antibodies is really a challenge. To avoid the limitations of the immunoassays, there's a high demand of alternative method to do as good as or better than the current HCP immunoassays is the mass spectrometry. The first one, they generally use the data-dependent mass spectrometry, also using the HPLC-based separation of phosphoprotein and identified and quantified proteins through mass spectrometry. But these are have a limitation of the fixed number of the precursors analyzed. The challenge to detect low abundant proteins. That's why new methods are developed from the data-independent mass spectrometry. They're still using SE-based separations, but they are able to identify, quantify individual proteins with more species. All ions with selected mass range can be covered, and also be minimal method development for target development, and also especially for the troubleshooting for process development and respectively can find a new target if there's anything happens. To establish the effective assay workflow for HCP monitoring, factors to be considered. The first one, as you mentioned, is dynamic range of protein concentrations, the product antibody versus the HCP proteins. Another one is the one heavily rely on the peptide library, the spectral libraries for the DIA method, especially the coverage and the quality of the libraries really affect the accurate quantification of the peptides, so real peptides or proteins in the products, and also the RT alignment. The sensitivity versus the reproducibility we need to consider from non-novel LC, capital LC, or microflow until to the analytical workflows. We need to see the balance. The quantification method, we need to use the relative or using the absolute quantitation, using spiking standard versus the peptides, using large or small proteins quantification will be slightly different. Sample preparations will be affected as well. The standardization and the automation should be also considered, especially for protein digestion and peptide recovery we need to consider as well. The QC for the accuracy and the linearity, producibility, and the specificity we need to also be considered for the LC-MS workflow. And also finally the throughput, robustness, and turnaround time and the cost per sample. This is one of the factors to be to consider. There are ways to deal with the dynamic range. First one is the enrichment of the host cell proteins before LC-MS. We have in-house tried one of the dilated the equal beads from Bio-Rad, the protein minor beads with the enriched proteins we got, purchased the drugs from the pharmacy. You see here, after enrichment, we can see many of the host cell proteins in addition to the major heavy chain and light chain, as well as some aggregates, as well as some depleting products. After LC-MS, we can see majority of the common proteins we can see. Also, there is sub ppm also, but we so confidently identified all these enzymatic proteins still remain in the product, although very low level. And another way to deal with the dynamic range is the proteomics strategies, the comprehensive fractionations. Here is the typical protocols using the 2D LC, the high pH and low pH LC, using online or offline to do in comparison with spiking of the different kind of proteins with the different fraction out of 5 to 10 fractions, and then online line team integrating this generally adapt. But this one already applied in general publications from Waters, and also Amgen are already using this strategy in the earlier stage of the MSCP characterization and monitoring. But this one takes a long time. One sample will take minimum 8 hours to 15 hours. This one really laborious can be really difficult to automation, and this one can be time consuming. Is not really good for the real time and online analysis of host cell during process development. That's why we need to have the one common method using the DIA method, using the single one-dimensional LC-MS run to get the comprehensive coverage of the host majority of proteins. One of the methods is a sequential window acquisition of all theoretical mass spectrum called SWAS. The SWAS is the one that you make use of the SWAS window, isolation window, using quadruple one. This is the small window to around in the 25 Dalton of the increment steps. Rapidly around the retention time, the steps cross the entire mass opportunity mass range within the available cycle time, usually around a few seconds, around 3 seconds, for example, cast all full scan composition mass spectrum for multiple analytes concurrently, resulting in a very complex MS spectrum of a high resolution fragmented data. After acquisition, the data files are interrogated with the proteins and peptides, using previously well-defined, determine the fragmentation patterns or spectral libraries. Extract ion chromatography for multiple fragments collected from the single analyte, or give high-quality peaks for accurate quantitation. Thus, the SWAS-MS has the features of all the precursors of fragmented MS can be acquired for the quantitation. can capture a complete digital archive of your samples in the first analysis, and also can detect low abundant analysis within the even in the complex samples, and also within one short time of the 1 algorithms. And within the Swashing method, the very important one is the peptide library. These are key points. To address this super library, we have really spent our efforts in collaboration with our partners, SAX. We comprehensively produced the build of the CHO assay library, since the CHO is the major key assay lines working on and the production of the monoclonal antibodies. We build the library from the one that's long or producing cell half the cell culture fluid and do digesting, as well as the cell lysate, the cell pellet. We separate different fractionations, the extractions, cytosolic fractions, and also membrane fractions, mitochondrial fractions, nuclear fractions. Get all different fractions to do the therapeutic digest, and then followed by offline basic high pH HPLC fractionation. Before inject mass spec, do DDA mass spec, we spec the index RT peptides. After acquisition of the peptides, we merge all the sequence results and build of libraries. And then this is the result. We use all different kind of the available search algorithm using the protein pilot, mascot, Peak Studio, protein discover, and also extend them all combined all these kind of search algorithms. So we have built up the library of most comprehensive CHO libraries, more than 11,000 proteins at 1% SDR, and every single protein group has more than two peptides. After we remove all the modified peptides and the shared peptides between each protein group, make sure we have the almost 10,000 unique proteins inside the libraries. To elaborate of these libraries, we have number of the studies. We use the HCP analyze using the downstream purification intermediate. We follow standard generic method, protein A and then 2 polish steps. We use the ELISA method from the 500,000 ppm to single-digit ppm with the platform standard ELISA assays. With the library, we can relatively quantify the more than 650 proteins in these libraries across the different steps, in purification steps, including clustering of the major common proteins with the minimal protein area 7,000, with the medium CVs less than 20%. We can monitor all the proteins using a Swashing method analysis. To identify, to quantify the proteins, we need to spec the proteins, standard protein with a lower amount to do the purification intermediate. You see it here. The intensity correlates well with the theoretical concentrations in different intermediate of the purifications. So for with the standard protein, we are able to quantify, absolutely quantify the whole protein in the different purification steps. This is the single-digit, different from the ELISA single-digit ppm, so we can identify individual proteins with the various steps. We can monitor the protein concentrations from very high to the very low, but still the tens of the ppms. Some proteins no change, some proteins but still remain some enriched some. But this one we need to monitor during the process of the downstream. So the linear range of detection, we also using the standard protein spectral IgG, the monoclonal antibodies with the purified one, and also in series of 5 times dilutions. You see here the linear range is very well present in the pure products, as well as the one with the more complicated one, the original materials. The original material will spec the proteins individually. The correlation was quite okay. So the spike in the more complex background still kept linear range is still maintained, the wide range. So the one of the reproducibility, is from different labs. Well established one of the landmark studies from the publication of the international 11 laboratories around the world using nanoflow LC coupled with the cube of mass spectrometries. The cross lab is using the HEC 293 cell digest with spiking with 6 orders of step isotope. They're showing with the intraday and the interday and every interlapse, they can identify more than 4,000 proteins with medium CVs around 22%. And also the limit range can reach to 4.5 order of magnitude. Shows the reproducibility, the Xuezhi method is really robust. And also, but the most, according to statistics by Agilent, the errors came from the sample prep, not the mass spectrometry, but it's also really robust. That we applied some of the in-house technologies using the filter-based digestion. Within 1 hour, we are able to see, using the 10 biological replicates prepared but with various levels of experienced users. We're using downstream. Other people deliver them of the proteomic sample preparation. We invite them to do the random study to test the sample prep is really robust or not. So based on the result, we saw the random samples of different groups with multiple runs. We get really in the lower range of change after total error sum. The normalization, simple normalization method, we can see the very consistently across the 10 people and 10 samples. This is using the Pearson correlation to show the every single cross-sample comparison. The linearity is really showing the reproducible across all the 10 samples. So to show the robustness of the host cell protein using DAA workflows, we have to using different LCs from the oxygen and the waters, and also we are showing them that still within the around the identical protein, the quantification of the total lysis around 10% CV. And also the while using different LC and different workflows, the flow rates up to nanoliters and also the capillary flow 5 microliter and the 50 microflows, all different kind of flow with compilation of the different mode of the sex machine 5600 and 6800. We can still see observe the CV within the range around 10% reproducible across the LCA system. With the different retention time, we will see here the nanoflow 6600 get a very good linearity, and also the capillary flow as well as the microflow. This one really linearity is the really satisfactory results. So I hope through this presentation I have already demonstrated the quantitative LC-MS complement the HCP ELISA method to while providing unbiased coverage and sensitivity, as well as the identity and the quantity of the individual host cell proteins. To characterization of the host cell proteins by mass spectrometry generates the invaluable information of the critical product attributes that could help in design a risk-based control strategy, especially in the future next generations will continue to process. This is really critical. DAA Xuezhi method supported by the comprehensive spectral library offers a fast, reliable, and robust sensitive quantification platform. Has the potential to support inline tracking HCPs, real-time biological development. Indeed, in the future direction, we are currently developing of the online analysis monitoring of the host cell proteins through directly sampling from the bioreactor, and the online with the downstream sampling as well as the analysis by Xuezhi method. I would like to take the opportunity to thank my staff in my proteomics groups, Lilian, Liu, and K1, as well as the Hui Dong, was working on hard work with the method establishment and library constructions, and as well as our downstream colleagues on Wei and Yuan Sheng from animal cell technology culture provided culture as, for our analysis. So last but not least, we need to thank the support from Stephen Tatum Sachs. As well, we also thank the funding support for our SR as well as Sachs. Thank you. If any question, I would be happy to answer.
Thank you, Dr. B. And now, as Dr. B just said, we will begin the question and answer segment of the webinar. But once again, I would like to remind the audience that you can still send your questions in via the questions tab located directly below your webinar screen. So now our first question. Why do you construct a CHO cell lysate full proteome spectral library instead of a CHO null cell HCCF?
Oh, okay. Thank you for this question. Indeed, it's very important. Indeed, we have already compared the HCCF, but since HCCF normally use null cells, every single time the different we have already written the publication or in our experience, the whole cell protein profile is different from the production cell as well as the null cells, and also strategically, all the proteins from the media circulating to HCCF supposed to be secreted or trafficking from the total host cells itself after either using secretion or lysing, releasing to the media. That's why we built up the comprehensive library from CHO, even from the membranes or the complete different fractionations. We already compared the HCCF library as well as the whole cell lysate. Even though using whole cell lysate can cover more than 80% of the whole cell proteins profile, and more so easily and this one no change can cover most comprehensive cover. Some special conditions with the HCCF, the one may change a little bit. That's why we can also build up the HCCF library in complementary to that one. We merged both together to still get the libraries working on. But so we are working on the most the consensus libraries, and currently we're merging the HCCF running more than 1,000 times. So collecting 1,000 times different conditions, the whole cell lysate after clearance. So we running that one. We build another libraries to see that one. Currently we see from the whole cell lysate library still cover more than 80% of the whole cell protein profiles and more robust.
Thank you for that answer. Our next question is there any database of well-characterized CHO HCPs that is available for targeted mass spectrometry analysis?
Okay, this is another good question. The one currently in the CHO database of the spectral libraries still not publicly available. That's why we also built our comprehensive libraries. All the libraries built by biopharma companies that don't share for outside. They build their own. They do the in-house analysis because they have the build process specific one. The profiles that I mentioned earlier, that the profiles of whole cell protein may be different. That's why nobody shares that one. Only recently, we just saw last year in February, released one of the libraries from using the thermal instrument HCD profiles. The one to identify protein in the collaboration of the Johns Hopkins University. But so far, using that library, we are not able to using for the quantitation yet. And that one because the library it didn't spec any retention time peptide, only for identification of proteins, not ready for quantification. That's why we built our own library for ready use the high quality and high resolution peptide ion libraries. We call it global assay libraries for working on the whole cell protein monitoring during the process development and also the final product release.
Thank you. And with that, we will conclude the question and answer session for now. But before I finish the webinar, I would like to ask our presenter, Dr. B, if you have any closing remarks or final statements.
Okay, so the whole cell protein indeed is a very important area for mass spec. Now, the one that is really needed to get indeed FDA is really or try to adopt a new orthogonal method to complement with current ELISA method. Either way to do, especially in the future direction of the bioprocessing, especially continuous process, we cannot really waiting for a long time, 3 to 4 hours of ELISA method to monitor the clearance. So this must have some alternative solutions to be on hand for real-time release tests and online analysis of host proteins during the continuous process especially.
Thank you. Now, I would like to take this opportunity to thank our presenter once more. That was Xuezhi B, Group Leader of Proteomics and Senior Scientist at Bioprocessing Technology Institute, A*STAR, for sharing his knowledge with us. I would like to remind our audience that you can view this and other webinars on demand by visiting biopharma-asia.com/on-demand-webinars. And if you are watching this on demand, please feel free to send your questions over to me at stephen.edwards@biopharma-asia.com. And we'll get those answers right back to you. Thank you, everyone. Have a great day.